High pressure is a method of choice for changing the relative population of a protein conformational sample. It is extremely useful for isolating and characterizing higher energy conformational states. Compared to other perturbation methods like pH or temperature, pressure perturbation is easy to apply and fully reversible. It is also local perturbation, primarily affecting regions with large cavities or void volumes.
To begin, introduce the nitrogen-15 labeled sample into the zirconia tube using a glass pipette, and ensure the sample seats at the bottom of the tube. To prevent the sample from mixing with the transmission liquid, overlay the sample with 200 microliters of mineral oil, and then fill the rest of the tube with transmission liquid. Put a single-use O-ring on top of the zirconia tube and slide the tube into the base. Then connect the tube to the high-pressure tether line and tighten the base to the cell by hand. Then apply 14.7 Newton meters torque to prevent leaks at lower pressure. To check the integrity of the pressure cell assembly, pressurize the tube up to 300 bars outside the spectrometer using cell support and containment vessel. After 15 minutes, reset the pressure to 1 bar and check for leaks with a clean, lint-free wipe. Insert the unpressurized tube into the spectrometer by carefully guiding the tether line. Slide the tube into the spectrometer until the tube reaches sample sitting position. Lock, shim, match, and tune the protium and nitrogen channels, and save the optimized shims for future. Set up transverse relaxation-optimized spectroscopy with hetero single quantum coherence spectroscopy, and proceed with recording a reference experiment at the atmosphere.